In this lecture, we'll be discussing photosynthesis, and this can get very complicated. So for this class, I just want this to be the basics, look more at big picture things and less on the real nitty gritty details. Okay, so I mean the really big picture here is shown on the title slide, this kind of cartoon version of it. Right? Photosynthesis is the process where plants, primarily plants we're going to be discussing, and some other bacteria, right? They use sunlight as an energy source to be able to take from water and carbon dioxide and turn them into oxygen and sugar. Okay, and that's what we're dependent on this planet, is the oxygen that they produce and the sugars that they produce. Right? And you can really think of this as all biological energy for the most part, at least for animals, comes from the sunlight in plants being able to produce oxygen and sugars. And we're going to discuss a little bit about, how exactly the plants are able to do this. So again, photosynthesis, it's the process of turning sunlight into energy, and in that process you produce oxygen from water, and you also produce sugars from carbon dioxide. And those are two separate processes there. You might have heard this before in other classes or from high school, right? You have a light reaction or light-dependent reaction, and what they call, I don't know if they still call it this, but they used to call it the dark reactions. Okay, so the light-dependent reactions, you need sunlight, as that name suggests, and those produce energy in the form of ATP and reduce NADP plus to NADPH. For the dark reactions, these are the reactions that involve the carbon dioxide assimilating, and you use ATP and NADPH to turn those carbon dioxide molecules into sugars, and that's in the Calvin cycle. So that's this part, the dark reactions, is what we'll be focusing on next lecture. Today we're going to be focusing primarily on the light reactions. And probably a better way to look at this is a actual diagram of what's going on here. So the light reactions uses light, takes water as the electron source, and through the chloroplast, this is where this is happening. All right, the chloroplast and the thylakoids, what we have happen here is water ultimately goes to O2, right? It's losing electrons there, it's being oxidized, and those electrons go to reduce NADP plus to NADPH. In that process, you're also pumping protons, which creates ATP. Okay. And then that energy is then used in the Calvin cycle, the dark reactions, to take CO2 to turn it into sugars. Okay. The dark reaction, it's kind of a bad name, and some people don't use that anymore because it doesn't require that it's dark. Okay, it can be light out, and you can still do those dark reactions. They call them dark just because you don't need light for them. Okay, right. But that's the two steps, or the two different cycles that we'll talk about here in the next two lectures. Right, and again, they do get very complex, and we'll try to focus on just the big picture stuff. Okay. This is the Calvin cycle, or the dark reactions, which again, we'll be talking about this in more detail next time, but this is a good overview of where we're going with photosynthesis. All right, so moving to section 1, let's talk about light absorption. Okay. So much like we talked about in glycolysis, we talked about substrate level phosphorylation. And then in chapter 19, we talk about in the electron transport chain in mitochondria, we talk about oxidative phosphorylation. In photosynthesis, what we have is photophosphorylation. All right, and that's where we're synthesizing ATP, but it's being driven by sunlight. And that's where that photo comes in. Okay. So we, again, just like in the mitochondria, we have electron flow through a series of membrane carriers, and we couple that to proton pumping. And H2O is the electron donor in this case, and by giving up its electrons, it turns into O2. And you remember from photosynthesis, that's the opposite of what happens, right? We have O2 being an electron, the ultimate electron acceptor. So in a way, you can kind of think of this as almost like running the electron transport chain in reverse in a certain way, right? NADPH is being formed ultimately, and in the electron transport chain in mitochondria, what we use as the electron source is NADH. So it is sort of like photosynthesis is kind of the, if you're looking at just the starting and end products, it is sort of like you're running it in reverse. And so here we have depictions of both. On the left side, this is the electron transport chain in mitochondria, right? And remember, we have this series of membrane proteins that have electron carriers in them. Excuse me, this is photosynthesis, it's green, right? The right side is the mitochondria. And we have, the series of electron carriers in the membrane. We get donor electrons, right, from NADH and also FADH2, depending on where you're coming in this chain. Those electrons are carried through those, and in process of doing that, you're pumping protons. You're pumping them from the matrix of the mitochondria into the inner membrane space. And then ultimately, your electron acceptor here is water, and it makes O2. Okay. These protons that you've created, a proton gradient around that inner membrane, and those are then used by complex 5, or also known as ATP synthase, right, to generate ATP. Well, it's very similar in photosynthesis, okay, except, right, we're doing some things backwards relative to the mitochondria. The first thing that's backwards, again, is you're using H2O as your electron donor now, not the acceptor. And when it donates its electrons, it turns into O2. Okay. The electrons are then carried through a series of membrane proteins. And by doing so, you're pumping protons. But now, the relative direction of the proton pumping is different. Okay, so this is the thicolloid membrane. We'll talk more about this, and we'll see more depictions later in this lecture. So don't worry so much now if you can't quite picture this or what these words mean. All right. The thicolloid membrane, and this is the thicolloid space, and then outside here would be the stroma. Okay, so we're inside of the chloroplast, and these are the little structures that are inside the chloroplast. Okay. Right. Ultimately, your electrons now go to NADP plus to make NADPH. Right. You're creating this proton, you're pumping them inside now of this membrane instead of outside like in the mitochondria. So your proton gradient is going to be from the inside. Okay. You're going to use that proton gradient again to produce ATP. But now you're producing it outside of this little structure. Whereas in the mitochondria, you're producing it ATP in the matrix. now you're producing the ATP in the stroma, in the chloroplast. Okay, so the direction is slightly different. In the acceptor and source of the electrons are kind of reversed, but they are very similar processes. And in fact, the complex 5 or ATP synthase is really almost identical in chloroplasts and in mitochondria. Right, so one of the things about this photosynthesis that's not really the best, if you just look at the overall process, water is a very poor electron donor. Okay, the reduction potentials, not really, you don't really need to memorize those, but has a very high reduction potential, which means it would be a very good electron acceptor. Okay, NADH is a good donor, right, because it has a very low reduction potential. Okay, so, but in this process, we're doing that in the reverse, right? We're using water as the donor and NADPH, or NADP plus, as the acceptor, right? So we're kind of doing this backwards in a way of what it really should be doing. Right, so you can think of the mitochondrial way as, you know, the natural way this would progress if you're just going by the reduction potentials. And how do the plant cells get around this? Well, that's where the light comes in. This energy that is absorbed from light is what allows this to happen in this direction. And we'll discuss exactly how that is a little bit later. Okay. So the flow of electrons, again, this is through a series of membrane-bound carriers, and you have things like cytochromes, quinones, and iron-sulfur proteins, just as we did in the electron transport chain in mitochondria. Okay. And again, just like in the mitochondria, this electron flow is coupled to pumping protons across a membrane to generate that electrochemical potential gradient, and that's what serves as the driving force to produce ATP. And this is all happening in chloroplasts. Okay, so the chloroplasts are these organelles where both the light-dependent and light-independent reactions, or dark reactions, are taking place. They're membrane-bound, and you have inside of them, I guess this is technically the chloroplasts here. When you have inner and outer membranes, and you have what's known as thycaloids, these discs, membrane-bound little structures that look like discs stacked on top of each other inside the chloroplast. Right, so the chloroplasts are somewhat similar to mitochondria. Right. The stroma is the aqueous phase enclosed by the inner membrane in the chloroplast, and that's sort of analogous to the mitochondrial matrix. Right. Although the overall structure of the chloroplasts and the mitochondria are different, the stroma you can think of as analogous to the mitochondrial matrix, because that's sort of the aqueous environment where a lot of these reactions take place. Okay. It contains most of the soluble enzymes that were required for the CO2 or the dark reactions. Okay, the thycaloids are the flattened discs that stack in the internal membrane system in the stroma. These are what contain the photosynthetic machinery. So all the photosystems in that the electron carriers are contained inside the thycaloids. Right. And then that term granule thycaloids, those are just where you have the thycaloids stacked connected in these stacks. The book talks about the Hill reaction, okay, and what the Hill reaction is specifically is just that the process of churning water into oxygen via using light, okay? So that's really all you need to know about the Hill reaction, right? You don't necessarily need to know anything about, these, what A is here, okay? Don't focus too much on this. In photosynthesis, A would be NADP plus, right? The ultimate electron acceptor. But here we have a dye, right? Hill reagent that you use. But okay, photosynthesis, this A is NADP plus. Right. So the point of the Hill reaction, right, is that it requires light. In the dark, you don't get any O2 development in this reaction, and your dye doesn't get reduced, right? And that proves that you need light for this reaction. Okay. And again, in photosynthesis and chloroplast, that A is NADP plus. It's what's accepting those electrons from water. So the overall process here is shown. All right, so we're going to talk a little bit about some fluorescence, and to do that, we need to talk about the electromagnetic spectrum and remind ourselves some of these terms. A photon, if you remember, is just a quantum of light, or one, you can kind of think of it as a light particle, right? Light can be both like a particle and a wave, but we use that term photon to mean one discrete energy packet or particle of light. Right? The greater the energy, the lower the wavelength. So if we look at just the visible region here, the highest energy is going to be here in the violet, although that doesn't really look violet on this picture, but the highest energy will be lower wavelength, so the violet end. Then if you keep going past that, you get to the ultraviolet. Right? And the lower energy is going to be more on the red end, right? And if you go lower in energy than that, you get to the infrared region. Okay, so just kind of keep those relative energies in mind here. When we're talking about light, oftentimes we use the Planck equation, and this is what gives us the ability to calculate the energy for any given wavelength or frequency of light. Okay? And wavelength and frequency are all are dependent, connected together, right, based on the speed of light. Whoops. Right, so we have the Planck equation is energy of the light is equal to Planck's constant, h, times the frequency. You can also write that in terms of wavelength. That would be Planck's constant times the speed of light divided by the wavelength. Right. And if you're curious, Planck's constant's there and the speed of light's there. They're both constants. Right. On an exam, those would be given if you ever have to use this equation. Yeah. We're going to talk about excited and ground states, right? So if you have an excited state, what does that mean? It just means that the energy of your, in this case, we'll talk about electrons, the energy of your electron is increased because it has absorbed a photon of light, right? By quantum mechanics, right, the energy of that photon is going to be equal to that electronic transition of going from a ground state to an excited state, a higher energy state. Right? And ground state, again, that's just stable state of energy, the lowest state of that, in this case, electron. Right, so what is fluorescence? Fluorescence is light emission accompanying the decay of an excited molecule. So you get an electron in an excited state. That electron goes back down to the ground state, and it releases a wavelength, a photon of light, okay, that release is always going to be a longer wavelength or lower energy than the light that was absorbed, just as you can imagine, by first law, it states that the energy of the universe is conserved. So if it was exactly conserved, you'd be releasing the same wavelength or energy of light that you absorbed. But real world, right? That's very rare where that happens. There's always going to be some losses, and those losses are primarily due to vibrational energy levels within that molecule. So you're always going to release a lower energy of light than you absorbed. So this excitation energy can be directly transferred. So if you excite something, an electron, into a higher energy level, it can be passed to another molecule through an excitation transfer. And what molecules are we talking about here? Well, we're talking about primarily chlorophylls. Okay, chlorophyll is sort of thinking of it as an antenna or a way of absorbing light energy. Okay. And particularly, we talk about them being green, and I'll explain that in a second too. Okay, chlorophylls are in the thylakoid membranes, okay, and what they have is this sort of porphyrin ring structure, and this should look pretty familiar if you think back to some hemes that we've looked at and hemoglobin from last semester. It looks pretty familiar, the ring structure here. The only difference really is you have a magnesium at the center instead of an iron. Okay. And then the other thing you have here is you have this long hydrophobic tail that's stuck on, and that's what allows it to stick in to the thylakoid membrane and kind of be bound in there. Okay. The difference between chlorophylls A and B, right? So this structure here would be chlorophyll A. Chlorophyll B, you just have this different group here. Right? Don't need to know that for this class, but that's just adding that different group, we'll see here in a second, changes what energy of light this molecule absorbs. So this huge molecule is affected by just what's at one little position here. There's also some other what are called accessory pigments in plants. So you might make sense, right? If chlorophyll, if you just have chlorophyll A and B, they absorb light at two very specific wavelengths. Adding some other pigments there allows the plant to absorb light at other wavelengths as well, so you're not wasting as much of the sunlight. Or if you think about it, at different times of day, the sunlight might have a slightly different color. These other pigments allow light to be absorbed throughout the day and at more areas of the spectrum. Okay. Do you need to know these structures? No, but things like beta carotene and lutein here are shown. And what do they have in common? They all have ring structures with at least one double bond in them. And then here we have these longer hydrocarbon chains that have alternating single and double bonds. Right, and then more complicated too, you have certain photosynthetic bacteria and algae that also have other pigments, and these other pigments actually have different colors, right? Don't really need to know them for this class. So if we look at the absorption spectra, chlorophyll A is shown here in purple, right? And you can see that it's absorbing a lot in the blue region, and over here in the red region as well. Right? And chlorophyll B is pretty similar, but now we've moved this peak in slightly and this peak in slightly. Right? So that one little change in that structure creates a fairly substantial difference in the absorption peak. But you can see that they together, you're absorbing pretty good chunks in the outer regions of that visible spectrum. And that's one of the reasons why these plants appear to be green, right? Because you're absorbing a lot of blue and a lot of this red region. So what's left, right? The colors that are left are what's reflected out, and those are more in the green region. Right? Some of the other pigments are shown here, like beta carotene here in orange and lutein here in black. Okay. These other pigments in the middle, those are those photosynthetic bacteria and algae that we don't really need to talk about, but they're absorbing, right, more in that green yellow region. So that's why they can appear to be different colors. Right, so this is where we get our action spectrum, and this is kind of the overall spectrum of where we're absorbing as plants. Right, you absorb a lot in that blue region, not so much here in the middle where it's green, and then again in that yellow to red region. Right, chlorophylls, they're absorbing this photons of light, and they're funneling that absorbed energy to what are called reaction centers. And they do that by excitation transfer. Very, very similar to fluorescence, but the molecules are so close together that it's not really considered fluorescence. Okay, what are terms here that are important? One is photosystems. We'll see that there are two photosystems. And what a photosystem is, we'll explain this in a second, but it's a functional array of light-absorbing pigments that's located in the thylakoid membrane. In them, you have what's known as a photochemical reaction center, and this is where that photochemical energy, the energy of a photon, is Right, so chlorophylls are acting as sort of like funnels. They take that energy from the photon, that absorbed energy, and they transfer it to reaction centers, and they do that by excitation transfer. So it's similar to fluorescence, but it's the molecules are so close together that you don't really consider it fluorescence. Okay, photosystems, they're the functional array of light-absorbing pigments that are located in the thylakoid membrane. And in them, they have this photochemical reaction center where this energy of the photon is transferred into a separation of charge. We'll discuss this here shortly. So how are these photosystems organized in thylakoid membranes? That's shown down here. And what you have are these squares shown here that are buried in this membrane. These are what are considered antennae. They ultimately absorb the light, and then they transfer this energy that was absorbed by that light, hopping through these antennas until they reach the reaction center in the center of these. Okay. So what are the antenna, what do they look like? Well, they're what these light harvesting complexes, or LHCs. They contain chlorophyll, all right, and they as well as other pigments. So here is shown lutein, and they're proteins that are kind of bound in this membrane. So how does this process work? Well, one of these antenna molecules, when they absorb light, what happens is that an electron from a ground state can be excited into an excited state, right? So you absorbed a photon of light. That energy equals the transition between the ground state and the excited state. So you get that excited state. And then what happens is this electron can then transfer that energy to the next antenna molecule, right? It transfers that energy, drops back down to its ground state. This electron in the ground state absorbs it and moves up to an excited state. So you get this sort of transfer of energy that way. And if these molecules were, antenna molecules weren't so close together, what that would look like is by dropping back down, this electron would give off a photon of light, right? So that'd be like fluorescence, okay? They're so close where you don't actually get that release of light. Right? And if you do that enough, what happens is this antenna, excited antenna molecule, again, can drop back down. By doing so, it excites an electron in this reaction center. Right? And do you need to know like all these separate steps and details? No, you don't. Just know the overall process. You get an excitation in an antenna molecule that can then be transferred via excitation transfer to the subsequent antenna molecules until you reach the reaction center. And what you're doing in this case is charge separation. Right? In the reaction center, this excitation energy is used to create a charge separation. And what you have here is really what you're doing when you're exciting that electron or electron pair in the reaction center. You're creating a negative charge, and the hole you leave behind is like a positive charge. Okay, so let's actually look into these reaction centers with some more detail. We're going to be focusing on plants, okay? And this is the schematic that explains how this happens in plants, these two photosystems. The book also goes into different photosynthetic bacteria that are very similar to this, but for this class, don't worry about the bacterial photosynthesis. It's actually just, it's a little bit more complicated, and what we'll focus on is the plants. Right. So this is called the Z scheme, and that's because this kind of looks like a Z, right? Kind of on its side, I guess. But it's showing the general transfer of electrons. Okay. I mentioned that there are two photosystems. They're called photosystem 1 and photosystem 2. The hard part or tricky part about this is that photosystem 2 is actually the first one that the electrons move through. Okay, so we're starting here from water. Water gives its electrons, and this is what's known as the O2 evolving center. Those electrons then go into photosystem 2, which has this what's called P680. And 680 stands for the wavelength of light that it's absorbing, right? So those electrons, light, cause it to become excited. When those electrons are excited, they can flow through these carriers. And the ones to really focus on are PQA and PQB. Those are plastiquinones. Okay? Those are very similar to ubiquinone. Right, they kind of have the same function as ubiquinone in the electron transport chain in mitochondria. Okay, so there are electron carriers mobile in the membrane that bring those electrons to, in this case, what's called the cytochrome B6F complex. Okay. You can kind of think of that as being very similar to complex 3 in the mitochondrial electron transport chain. Okay, they then go into a molecule known as plastocyanin or cyanin. Okay, this is very similar to cytochrome C. It then moves those electrons to photosystem 1, okay, in the P700 reaction center. You can kind of think of this now as, well, I shouldn't. So this P700, right, that 700, that's a longer wavelength or lower in energy than 680, right? Remember, you go from, if we're thinking about fluorescence here or energy transfer, you tend to go from higher energy to lower energy, okay? So that's why this is a bigger number or lower energy, okay? So light now, you can excite that. Those electrons then get transferred through some other carriers. Okay. And we'll talk about this too here in a second. In photosystem 1, you can go linear. The linear transfer, you go into ferredoxin NADP plus reductase, which ultimately donates them to NAD plus to make NADPH. In the absence of that, NADP+, you still want to be making ATP and you still want these electrons to flow. So then you can have a transfer back to a plastoquinone from ferredoxin. And this would make it cyclic. So it just kind of goes in a cycle, right? Which might seem a little silly, but it's to generate ATP. In the absence of NADP+. So NADP+ can be limiting sometimes, and in which case you still want to be able to make ATP, and that's a workaround. As I mentioned, this is very similar to photosynthetic bacteria. And here we have, and the book shows purple bacteria and green sulfur bacteria. So you can think of photosynthesis in plants as being kind of a combination of those two. And ultimately, endosymbiotic theory, probably what happened is, right, a eukaryotic cell at some point absorbed purple bacteria and a green sulfur bacteria or something that had a combination of the two already and introduced that machinery into that eukaryotic cell. Okay, one thing I haven't mentioned yet that I probably should have is that plant cells do have both mitochondria and chloroplasts in them. So they're doing both of those processes and have the ability to do both of those processes, unlike us, which we only have mitochondria. Okay, so the first photosystem that's used in this process is photosystem 2. Okay, so photosystem 2, we could describe it as a pheophytin-quinone type system, like the purple bacteria photosystem. Okay, it contains approximately equal number of chlorophyll A and B. Electrons pass from the excited P680 special pair to the cytochrome B6F complex. Okay, the electrons that are lost from this P680 are replaced by electrons from H2O. Okay, the second photosystem is photosystem 1. This is structurally related to that green sulfur bacteria. Okay, it contains a high ratio of chlorophyll A to chlorophyll B, and that's why the absorption number is just a little bit different there. Okay. Again, it has two paths. Linear path, the excited electrons through this photosystem go to NADP plus. The cyclic method, those electrons go from ferredoxin to plastoquinone, again through the cytochrome B6F complex. Our net equation for electron flow from H2O to NADP plus, right? We've sort of saw this before, but we didn't have the number of photons in there. So we have two waters have to be oxidized to make one O2 molecule, right? And by doing that, we get two NADP pluses going to two NADPHs. And we have a total of 8 photons that are required for this process. So that's just some bookkeeping to keep in mind. Okay. This is kind of a cartoony structural depiction of photosystem 2. And we have some more detail, crystal structure type detail here in the reaction centers themselves. What you'll notice as well is you have this manganese calcium oxide complex. This is what's going to be the O2 evolving complex, or at least part of it. Okay. And what we have is the lumen side and the stroma side. So the lumen is inside the thicolloid. The stroma is going to be the outside of the thicolloid. So now if we look in a little bit more detail of actually what are these electron acceptors, okay, we start with that P680. Okay. You have an electron passed from the excited P680 to what's known as pheophyton. Probably don't care that you know that term, pheophyton. But then that pheophyton donates to plastoquinone. And that's the important one to remember, plastoquinones. So you have plastoquinone A and plastoquinone B. Okay. The electron removed from P680 is replaced by an electron from water. Okay, so that's where we get water. And how that works is you have this oxygen evolving complex where you have this manganese calcium oxide coupled to a tyrosine, right? So when you get an electron moving from P680, that creates a hole, and that's what gives the driving force for that hole to be filled with electrons from water. Okay, so you can kind of think of that as like a semiconductor in a way, where you have holes, and those holes act as positive charges, and you need the electrons moving to fill them. Okay, the overall reaction initiated by photosystem 2 is shown here. Okay, you would have the stoichiometry here. You have 4 P680s. You need to reduce 2 plastoquinone, excuse me. And that takes 4 photons. Okay, photosystem 1, here's a picture of it shown in much greater detail for some reason. The book doesn't, I guess they want to give us as much detail as possible here, and not for photosystem 2. The reaction center here, again, is P700. It was P680 in photosystem 2. In photosystem 1, it's P700. Okay, so how this works, and this is probably a little bit easier to see, right? Remember in photosystem 1, it gets its electrons from a plastocyanin. Okay, that mobile protein carrier is sort of similar to cytochrome C. The electrons now are donated into P700. They go through some other cofactors, what's known as phyloquinone, this Q here. Don't really care that you know that. But then they go through 3 iron-sulfur clusters. Again, that should look similar to our electron transport chain in mitochondria. And ultimately to this protein called ferredoxin. If you're curious, the plastocyanin, it's a soluble, sort of like cytochrome C, right? Soluble transfer protein. It actually has a copper. It contains copper, right? Cytochrome C had a cytochrome with iron. Plastocyanin uses copper. So ultimately what happens here is ferredoxin is going to be used to transfer electrons to NADP+. And it does that through this ferredoxin NADP+ reductase. So that's the 4th electron carrier in this chain, which ultimately transfers those electrons from reduced ferredoxin to NADP+. And that overall reaction, you would need 2 reduced ferredoxins to reduce 2 NADP+. The link between the 2 photosystems here is cytochrome B6F complex. Okay. It contains, as the name suggests here, a B-type cytochrome that has 2 heme groups. It has these RISC iron-sulfur proteins and also a cytochrome F. Okay, so not really something that you need to know, but that's where this name is coming from. Okay, and again, it's analogous to complex 3 in mitochondria. The electron flow through this complex produces the proton gradient. So it's one of the ways in which this proton gradient is being produced by electron flow. Okay. So here we have of this complex, sort of a more cartoon, I should say a more detailed structure of the cofactors are shown here. Right. Ultimately, we're going from plastoquinone to this plastocyanin mobile carrier protein. I think this one's a little bit easier to see. It's a cartoon depiction, and it should look very similar to complex 3. You even have a Q cycle here, and you have a BH cofactor and a BL cofactor. So it's very similar to complex 3 in that regard. Do I care that you know this Q cycle here for this? No. That Q cycle is more important in complex 3 in this class, but just to point out that it does have this Q cycle, right? It's very similar to complex 3. Right? And ultimately what you're doing here is you're taking 2 protons from the stroma side and pumping them into the thicoid side or lumen side. Okay. And we mentioned this before, there's 2, photosystem 1 can operate in a linear fashion or a cyclic fashion. So the linear fashion of photosystem 1 transfers to NADPH, so you're creating NADPH and ATP by proxy. Okay, the cyclic electron transfer, you're producing only ATP, you're not producing NADPH. Okay, this allows plants to adjust the ratio of ATP to NADPH to match its needs. You can also think of it as if there's no NADP plus around, right, that can be limiting in this process, you can still produce ATP. So in cyclic photophosphorylation, you're just converting ATP to ADP to ATP, and that's accompanying this cyclic electron flow. Okay, and you can kind of think of that, it's sort of like a feudal cycle. The difference is you can keep this cycle going because you're using the absorption of light, that energy from the absorption of light, to just keep it going, right? So you can just keep, in a sense, as long as there's light there, you can just keep absorbing light and using that energy to keep this going forever, okay? Right, and really a very simple overall reaction, right? You're taking ADP and you're making ATP and you're using light to do it. If you're curious about where these are in how they're set up in the thylakoid membrane, that's shown here. Okay, so you have these light harvesting complexes, or antenna, you can kind of think of them as, interspersed between photosystem 1 and photosystem 2, and then your cytochrome B6F complex as well. Okay. And coupled to that, you also have your complex 5 or ATP synthases. Okay. Don't need to know that. You know, I'm not going to ask you on an exam to draw this out and put their locations. Just know that they're all in the thylakoid membrane. Okay. So going back to the start, how do we get these electrons, right? This is the first step in the whole process, but how do we get them, right? Water is the ultimate source of these electrons. So for every 2 water molecules, you can make an O2, right? By doing that, you give up 4 electrons. Right? And this is done at the oxygen evolving center. Okay, it's going to pass 4 electrons, one at a time, to P680. Okay? And P680 plus or P680 star, that indicates that it's in that excited state. It has absorbed that photon of light energy, and that's moved to an excited state. And it can only accept one of these electrons at a time. Right, so the overall process here, we have 2 waters coming in, right, and that's going to produce 1, 2, 3, 4 electrons. But again, these have to go one at a time. Right. What's shown here is that you have a tyrosine residue that helps with this electron transfer. So this tyrosine is the immediate electron acceptor from water. It loses an electron and a proton to generate a free radical. Okay, probably not super important that you note that. That tyrosine radical then oxidizes this manganese complex in the oxygen evolving center. So the tyrosine oxidizes that manganese complex, and that's what gives us this driving force to take electrons from water. So just a quick question to make sure we're all on the same page. How many photons does it take to produce 1 molecule of O2 through photolytic cleavage of water? Right, and remember that's 4. Okay, it takes 4 photons to produce 1 molecule of O2. And by doing that, you're also generating 4 electrons. So this oxygen evolving center is acting as a proton pump driven by electron transfer. Okay, so 4 protons produced in this reaction are released into the thylakoid lumen. Okay, so this is another, we saw that in the cytochrome B6F complex, it's pumping protons. This oxygen evolving center is also acting to pump protons. Okay. So our overall reaction initiated by light in photosystem 2 here, we have P4P680s plus 4 protons plus our 2 plastiquinones plus 4 photons of light goes to 4 excited state P680s plus 2 reduced plastiquinones. And the overall reaction of water splitting in photosystem 2, we have 2 waters plus 2 plastiquinones, oxidized forms, plus 4 photons, goes to O2 plus 2 plastiquinones reduced. The oxygen evolving center, if you're curious, it's details of its structure are shown here. Not going to really ask you for this. The important things to note about it are it uses manganese and calcium, but most importantly in here are the manganese, because that's what can be oxidized and reduced. It's coupled to a tyrosine residue, right? So that's another important thing to remember about it, but the actual shape of it, not so much. Okay, last section we'll talk about today is section 3, and that's how this is all coupled to ATP synthesis. Right, so we have our photosystem 2 and photosystem 1 that absorb light. By doing so, they're creating a proton gradient here on the inside, the lumen side of the thycaloid. Right, those electrons that are generated from water, right? Water gives up electrons to make oxygen. Those electrons that are generated here flow through the cytochrome B6F complex. That also creates a proton gradient. Okay. Ultimately, the electrons go through photosystem 1 to ferredoxin and into NADP plus to make NADPH. Okay, so the energy to do that, to take electrons from water to make NADPH, that energy is all given by light. Okay, doing this process creates this proton gradient. That proton gradient is then used to create ATP. Okay, and again, this is almost identical to complex 5 or ATP and synthase in mitochondria. One major difference, though, here is you see that the gradient's being generated in the lumen, okay? And that is then pumped out into the stroma where the ATP is generated. Okay, so it looks slightly different than the direction. If you're thinking of this as the inner mitochondrial membrane, looks slightly different, sort of backwards direction compared to the mitochondria. The lumen, sometimes known as the P side or positive side, because that's where the proton gradient is being generated, and the stroma is also sometimes called the N side or negative side. Okay. So approximately 12 protons move from the stroma into the thycaloid, the lumen side of the thycaloid, for every 4 electrons that are passed through this machinery. Okay. The delta G equation here, don't really care. It's not something we're going to be tested on this class, but if you calculate that, you get negative 17 kilojoules per mole of protons. And experimentally, the measurements show that 8 photons drive the production of 1 oxygen. Okay, so stoichiometry, you need 8 photons of light to produce 1 oxygen molecule, and that process yields about 3 molecules of ATP. Okay, so sort of weird numbers, right, to remember. 8 photons to 1 oxygen, and that yields approximately 3 molecules of ATP. Unfortunately, you know, they're not simple ratios, but that's the basic stoichiometry of photosynthesis. So about how many ATP are generated from the oxidation of H2O to 1 molecule of O2, right? So that'd be 2 molecules of H2O to 1 molecule of O2, right? That would be 3. So the overall reaction for linear photophosphorylation, we have 2 H2Os, 8 photons, 2 NADP pluses, about 3 ADPs, and about 3 inorganic phosphates, and we get 1 molecule of O2, 3 molecules of ATP, and 2 molecules of NADPH. This is the the overall reaction that I'd like you to remember. If we compare this to the mitochondria, how are they similar? Well, if you look at the mitochondria, we're moving electrons through the electron transport chain, and we're doing that to generate a proton gradient in this darker orange colored region, the inner membrane space. That's the P side in this organelle. And then that's used to generate ATP in the matrix, so the lighter orange. That would be the N side. Okay, the chloroplast, you know, I think of it as kind of a backwards of the mitochondria. The thycaloid, the P side, this darker colored region, that's on the inside of the thycaloids here, which are, I kind of think of that as the center of the chloroplast, but it's really analogous to this inner membrane space here in the mitochondria, right? Because that's where we're creating the proton gradient. That's the P side, right? That's being pumped out to generate ATP in this lighter green region, the stroma. Okay, so they're very, very similar processes. And ultimately, in this class, I think that's the most important thing to note about photosynthesis is just how to compare it to the mitochondria and be able to list the similarities. Right, and so essentially it's identical to a mitochondrial analog, a chloroplast, right? Knowing how to describe that in, you know, on an exam, being able to match the different processes or the different machinery from the two would be what to know. We skip over a lot in photosynthesis in this class, and that's the bacteria and the algae. And I just wanted to mention this at the end of this lecture. These bacteria are actually the first things that came up with this ability to do photosynthesis, and it happened before there was oxygen on the planet. And so they're really what are responsible for our planet having this oxygen-rich atmosphere, which we use to breathe and to do respiration, right? So this occurred, they think, about 2.5 billion years ago. Cyanobacteria acquired the two photosystems that could operate in tandem. And that's what allowed the ability to split water and release oxygen into the atmosphere, and thus create an oxygen atmosphere. And then, subsequently, plants and animals could evolve using that oxygen atmosphere. Okay. And there's some other photosynthetic microorganisms that can acquire electrons from other donors. And so, not just water, you can use things like hydrogen sulfide, or even lactic acid can be used to do this, and some other photosynthetic microorganisms, which we just don't really go through in this class in much detail because, it gets quite complicated, but I'd be amiss if I didn't mention them because they are, at least at certain points in the evolution of our planet, were very important and influential. Okay. So that wraps up this lecture. Next lecture, we'll be talking about the dark reactions or the Calvin cycle. And again, we'll probably just go very, very general in that and not really go into a whole lot of details with it, at least for the purposes of this class.